Good day students, myself Rajendra Shetty, Assistant Professor at Ganpar Parsikar College of Education, Harmal, Goa. Title of the unit is Primary Structure of Organs. Module name is Structure of Monocot Stem. In this module, we are going to study tissue system of monocot stem, ground plane of tiers of young monocot stem, and enlarged cellular representation of tiers of young monocot stem. At the end of this module, we will be able to understand arrangement of different tissue system in monocot stem and know its anatomical characteristics. When we look at the tissue system of monocot stem, we see that it consists of three different types of tissue systems. First one, epidermal tissue system. Second, ground tissue system. And third, vascular tissue system. Epidermal tissue system consists of epidermis. Ground tissue system consists of ground tissue. At ground tissue, we see some layers of hypodermis, but no any differentiation of general cortex, endodermis, pericycle, pith and pith rays are seen. Vascular tissue system consists of vascular bundles. These vascular bundles in monocot stem are scattered in ground tissue. When we look at the ground plan or outline of tiers of young monocot stem, here we have taken up example of a young meristem. We see outermost layer as epidermis. Epidermis is followed by cortex. Cortex shows few layered hypodermis and rest of the tissue is ground tissue. Stele is a tectostelic type. In this stele, vascular bundles are scattered in the ground tissue. Stele does not show differentiation as pericycle. It does not show differentiation of pith and medullary rays. When we look at enlarged view of the section of monocot stem, we see outermost layer is epidermis. It is single layered. It is made up of compactly arranged, thin walled, barrel shaped parenchymatous cells. These are living cells. On its surface, we see presence of cuticle. This cuticle is a waxy coating of cutin. Being it is having this waxy coating, it prevents loss of water of inner tissues of stem and also gives protection against mechanical injury, high light intensity, pathogen attack, etc. No any epidermal hairs or trichomes are present on the epidermis of monocot stem. Sometimes we see presence of stomata. The next layer is hypodermis. Hypodermis is made up of few layers of compactly arranged sclerenchymatous cells. These cells are thick-walled and lignified. Because of presence of this lignin, it provides mechanical support to growing stem. Hypodermis is followed by ground tissue. In ground tissue, we don't see any differentiation of general cortex, endodermis, pericycle, pith and pith rays. This is one of the striking difference what we see between a monocot stem and a dicot stem. In case of dicot stem, there was clear, distinct general cortex, endodermis, pericycle, pith and P3s, which we don't see in this monocot stem. The cells are thin-walled, parenchymatous cells with large number of intercellular spaces. The main function of this ground tissue is storage of food. When we look at the vascular bundles, we see that these vascular bundles are scattered in the ground tissue. This is again a striking difference between a dicot stem and a monocot stem. In case of dicot stem, we saw vascular bundles were arranged in a ring form at the periphery of the pith region. Here, we see them scattered throughout the ground tissue. When we look at the arrangement of the vascular bundles, what we notice is in the central region, they are less in number and widely placed and larger in size. Whereas, when we look at the peripheral region of the ground tissue, they are more in number, compactly placed and smaller in size. When we look at each vascular bundle, we see that each of these vascular bundle is nearly oval in shape. Each vascular bundle is enclosed by sclerenchymatous bundle sheath. The bundle sheath is the vascular bundle is conjoined, collateral and closed, meaning xylem and phloem are present side by side. Mainly, phloem is towards the periphery and xylem is towards the inner side. Xylem is Y-shaped. Two metaxylem vessels with wider cavities lie at the two lateral arms, and protoxylem, usually one or two, with narrow cavities lie at the base. This type of arrangement is not seen in case of the dicot stem. Also, there is presence of lysogenous cavity containing water. It is present just below the protoxylem. Phloem lies outside the xylem and is comparatively smaller to xylem tissue. It is composed of only sieve tubes and companion cells. Phloem parenchyma is absent in phloem. To summarize, we see epidermis, in which cuticle is present but no any epidermal hairs are present. Hypodermis is sclerenchymatous. Ground tissue does not show any differentiation. Stele is a tectostele. Vascular bundles are scattered in ground tissue. They are conjoined, collateral and closed. Xylem is endark and Y-shaped. Phloem parenchyma is absent. These are the references. Thank you.